Hey guys, today we're going to be talking about chapter 7 from Campbell Biology in Focus, and this is going to be over cellular respiration. So we kind of skipped this and went ahead to photosynthesis, but now we're going back to chapter 7. So we've already talked about photosynthesis and all of those reactions. Now we're going to talk about cellular respiration because this occurs in all organisms. So I put this picture in here. I added this into the presentation to give you an overall image of what we're going to be talking about today. So there's three main stages of the process of cellular respiration. If we're talking about aerobic cellular respiration, there's glycolysis, there's the citric acid cycle, and then the electron transport chain. Those are the three big steps. This is a really good picture I need you to come back to whenever you don't understand something or you need the overall image. I also really like that it's telling you like the investment stage versus the payout stage, so you can see our net gain overall. Okay, so I need you to come back to this picture throughout if you're confused about the overall process. And I also need you to come back here after you're done listening to make sure that you can explain these steps, because then you know it for your test. Okay, so we know all about energy from ecology, freshman year biology, okay? But we know that energy flows into an ecosystem from the sunlight. That's our solar energy. And we know that the sunlight, now that we just talked about photosynthesis, goes into the leaves of our plants. It goes into the chloroplast, excites the electrons. We have the plant making chemical energy out of this solar energy. So we have a transfer of energy from solar to chemical. All the time, everywhere, the process is identical, if you remember your rap song. Okay, so photosynthesis in this process is going to generate oxygen, and oxygen and organic molecules like glucose. Remember that organic molecules just means that it contains carbon to hydrogen bonds. C6H12O6 definitely fits that bill. Okay, so these are going to be used as fuel for cellular respiration. It's a cyclical relationship, and that's the very last slide of this presentation that I also added in for you. Photosynthesis and cellular respiration definitely rely on each other 100%. One cannot exist without the other. Okay, so basically we already talked about photosynthesis and how it's going to make oxygen and organic molecules called glucose. And now we're going to combine those two things in order to enter into the cellular respiration process. So our cells are going to use this chemical energy that's stored in our organic molecules like glucose in order to generate ATP, which powers work. So ATP is kind of like a currency of our cells. You know that the mitochondria is the powerhouse of the cell. If you learn anything about biology ever in your life, that's probably the thing that you remember the most. It's kind of become like this awful joke, right? So this is where we're going to be focusing today, all of our energy in the cell, literally, because it's generating ATP. So we're going to be talking a lot about the mitochondria today. We're going to be talking about how to create ATP. That's the whole point of cellular respiration. Like, why do you need to do it? Because you need ATP. Why? Because it's energy. Without energy, you're dead. That's how it works. Okay, so previously when we talked about photosynthesis is an anabolic reaction, which means it's a building up reaction. Think about anabolic, like anabolic steroids. I like to pick things up and put things down. We're talking about bodybuilders making big muscles. Catabolic is exactly the opposite. So instead of making glucose, now we're going to be breaking glucose. And here's how I like to remember it. Catabolic, cats. I don't like cats. I have dogs, because they bark in every one of my videos. Catabolic, cats. Cats have claws. Claws can tear things apart. Catabolic, cats have claws to tear things apart. Okay, so this is a breakdown. This is a breakdown of organic molecules like glucose, and it's an exergonic reaction, which means it's going to be releasing heat or energy. Okay, so there's two main types we're going to talk about, and it's kind of reiterated in the next few slides, but fermentation is one of the types, which is just a partial breakdown of the sugar, and it occurs in the absence of oxygen. So if there's no oxygen present, fermentation. Okay, and then aerobic respiration, aerobic refers to oxygen. So this is going to consume organic molecules and oxygen, so combine them together in order to create ATP, which is a much more efficient process. So anaerobic respiration and aerobic respiration are similar in that they both consume compounds other than oxygen. So like I said, there's two main parts. You have either aerobic or anaerobic. So aerobic is again referring to oxygen. So this occurs in the presence of oxygen. It's a much more efficient process, whereas anaerobic occurs without oxygen. And it's kind of like a last resort for like us as humans, but other organisms like rely on anaerobic respiration, like that's all that they do. And that's how their little cell bodies are created, right? Okay, so we eat things, carbs, lipids, proteins. We eat these things. There's 3 different classes. Okay, yes, the 4th class is nucleic acids, but we don't think about that like a macromolecule for us that we're consuming, right? So carbohydrates, fats, proteins. You consume all of them. They all give you fuel, right? It's not like, oh, if you eat a stick of butter, you're not going to get any fuel. I mean, it's just a different kind of fuel than if you were eating a donut, which contains a lot of carbohydrates. But we're always going to talk about glucose going through this process. Just because it's the most fluid coming from cellular respiration, from photosynthesis into cellular respiration, glucose is just like the ideal molecule to follow because it doesn't, quote, break any of the rules. And it goes through the complete cycle of cellular respiration instead of getting to like skip steps, which if you consume fats and proteins, you don't start in the same place in the cycle. It's a cycle, like a circle. You just enter at different points in that circle. The same process happens, but I don't want to start off telling you maybe about like a protein that gets to skip a couple of steps because then you don't learn those steps. So that's why we always talk about sugar, glucose, right? But do you eat other things? Yes. Do they give you fuel? Yes. We just talk about glucose because it goes through the entire process and it's coming fresh out of photosynthesis. So this reaction is happening in plants. So as soon as the plants make this glucose, well, it's just like your neighbor handing you a cup of sugar. Great, thanks, Linda. Now what do I do with it? In order to get energy, you got to break it down, right? So the plant has glucose. Great, now you got to break it down. You spent all that time building it up. Now you got to break it, right? So cellular respiration is really cool because it happens in every single organism, whether it's aerobic respiration or anaerobic respiration. But like I said, that's why we use glucose. Yes, you get fuel from other sources, but this is just the one that goes through the entire system. And like I said, it's coming straight out of photosynthesis. We just talked about this, so it's going to make better sense this way. So redox reactions kind of drive this whole process. And we talked about that with photosynthesis as well. So when we're talking about redox reactions, we're talking about oxidation and reduction happening together. It's like coupled reactions. Okay, so it's a transfer of electrons during our chemical reactions that release energy stored in organic molecules like glucose, because that's what we're talking about, right? So this released energy is ultimately used to create our ATP. That's the whole point of cellular respiration. If that's like the takeaway that you get from all of this, then you learn something. Cellular respiration is used to make ATP, period. That's what it's used for. Why do we need it? To make ATP. Without ATP, you're dead. Okay, so redox reactions, like I said, they're coupled reactions. It's chemical reactions that are going to transfer electrons between reactants, and they're called oxidation reduction reactions, or short redox reactions. Now, remember I gave you a little acronym, OIL, RIG. Oxidation is loss, reduction is gain. So this is what it means. So in oxidation, you have a substance that's losing electrons. Oxidation is loss. OIL, O-I-L. Oxidation is loss. Okay, these things are oxidized. Okay, in reduction, RIG, oil, rig, this is the RIG part. Reduction is gain. A substance gains electrons. So if you just remember oil rig, it'll help you remember which is which. Okay. So in reduction, a substance gains electrons or is reduced. So the amount of positive charge is reduced. So here we have an example of a redox reaction. Okay, so you have Na that becomes Na plus because it is losing electrons, because it's becoming more positive. You know that it's oxidized. Oxidation is loss. Remember, electrons are negatively charged. So if something gains a positive charge, it had to be losing electrons. And the opposite is true for our Cl, like it's a Cl minus here. So this is reduced because it's gaining electrons. Remember, electrons are negative. So because we throw on that negative sign there, that minus sign, we know that it's actually gaining electrons. And again, that's oil rig to help you remember all of that. Here's another example. If you have a minus and it goes away, you've got to be getting rid of electrons because you're becoming more positive. That's oxidized. The opposite is true on the other end here. If you're adding a negative, then you know you're gaining electrons because electrons are negative. Okay, so the electron donor, the thing giving up electrons, is called the reducing agent. And the electron acceptor, the thing that's taking on some of the electrons, is called the oxidizing agent. And that goes back to our acronym, OIL, RIG. Make sure that you can differentiate between those. So some redox reactions do not transfer electrons, but change the electron sharing and covalent bonds. Remember that covalent bonds are how electrons kind of create like a little bridge between our atoms in order to share electrons to complete their valence electron cloud, right? An example of this would be methane and oxygen. So here's kind of what this looks like. So we have our methane is our reducing agent, our oxygen, which remember occurs as a O2 molecule. It's our oxidizing agent. Makes sense because it's oxygen. It's like they did that on purpose, right? So we have our methane that's becoming oxidized. It's literally gaining oxygen, okay? And then we have our oxygen that's becoming reduced because it's gaining electrons in the form of hydrogen. And you can see all of the chemical equations or all of our little dot structures spelled out at the bottom of the screen for you. Okay, so our redox reactions that move electrons closer to electronegative atoms like oxygen release chemical energy that can be put into work, right? So anytime that you're releasing energy like heat, it can be used to do work. Okay, so oxidation of our organic fuel molecules during respiration. Remember I said that photosynthesis is generating our oxygen and it's generating glucose. Now we're going to take these organic molecules called glucose and we're going to mix them with oxygen in order to oxidize them, and we're going to be breaking them down. Okay, so during cellular respiration, the fuel, glucose, because that's the ideal fuel, is oxidized. And oxygen is reduced. These redox reactions are going to be very important that you understand what they mean. Remember oil rig. Okay, so organic molecules with an abundance of hydrogen, like carbohydrates and fats, are excellent fuels because they have a whole lot of this hydrogen that we can use to reduce things. And as hydrogen with its electrons is transferred to oxygen, energy is released, and that can be used in ATP synthesis. And what's the point of cellular respiration? That's right, it's to make ATP. Hopefully that's what you said. I'm going to believe that that's what you said because you're smart and you know this. That's great. Okay, so here's what's happening there. So we have C6H12O6 called glucose plus 6 O2 molecules called oxygen that becomes carbon dioxide, CO2, water, H2O, and energy. Okay, so if you look at this equation, you'll notice that it is exactly the opposite of the photosynthetic equation. It's just the same thing backwards. If you can remember, plants take water, sun, and CO2 to make glucose and oxygen. Flip it, and now, boom, suddenly you know the cellular respiration equation. It's very convenient. But no matter how bright you think you are, that energy is not going to be sunshine coming out of you, okay? It's called energy, ATP is what we're talking about here, okay? So we have glucose that we're mixing with oxygen because we eat things and we breathe. So we're getting glucose and we're getting oxygen. And we're turning that into CO2, comes out in your little hot breath, right? Water, I'm not talking about like you produce a whole bunch of water, but if you go on a mirror, it fogs up, not because your breath is hot. I mean, maybe, right? But then it's also turning the mirror foggy because of the water vapor that is coming out of your mouth, right? And then energy is in the form of ATP because what's the point of cellular respiration? To make ATP, you're correct. Okay, so here you can see that our glucose molecule, C6H12O6, says it becomes oxidized. Okay, so oxidized, OIL, oil. Oxidation is loss. We're removing electrons here because we have lost our hydrogens. They're gone, right? And then with our oxygen, our 6 oxygen molecules, they become reduced. Reduction is gain, RIG, right? And we're adding in electrons there in the form of hydrogen. So it's a redox reaction that's occurring. Okay, so we're going to go over the stepwise energy harvest of NAD plus and the electron transport chain. Okay, so there's an electron transport chain in photosynthesis, and there's also an electron transport chain in cellular respiration. So in cellular respiration, we have glucose, which is the main thing that we're talking about, and other organic molecules, that could be anything else that you're eating, that are going to be broken down or catabolized, broken down, a catabolic reaction in a series of steps. And it's a lot of steps. Okay, so the electrons from organic compounds like glucose are usually first transferred to NAD+. Notice that there's no P here. NADPH is in photosynthesis. That's where I like to think about the P coming in. We're not talking about photosynthesis, so there is no P here. Okay, so electrons from our organic compounds, glucose, are usually first transferred to NAD+, which is a coenzyme. It's just something that functions with the help of an enzyme. So as an electron acceptor, right, because it's going to be accepting those electrons, NAD+ functions as an oxidizing agent during cellular respiration. So for each NADH, which is the reduced form of NAD+, because it has gained electrons, represents stored energy that is tapped into to synthesize our ATP inside of the mitochondria. Okay, so for each NADH, that has stored energy in it that we can use to then create ATP. So NADH can be turned into ATP. NAD+ does not have enough energy to do that yet. We have to add electrons to it by reducing it. So this is what NAD+ looks like, okay? And then when we add hydrogen to it from your food, then it becomes reduced, okay? And that's going to create our NADH, which then has energy that we can turn into ATP. So NADH passes the electrons to an electron transport chain. And unlike an uncontrolled reaction, the electron transport chain passes electrons in a series of steps inside of one explosive reaction. Instead of one explosive reaction. So if you imagine like a bouncy ball going down or like a slinky going down a whole bunch of stairs or something, you're kind of like changing the energy state. We looked at this when we looked at photosynthesis. So you guys already know that. The electrons are getting passed through different photosystems and photosynthesis. And here it's just different enzymes that they're passing through. And each time they're going into a lower and lower energy state. So you're not just having this huge like combustion reaction, you're having a series of a whole bunch of little tiny reactions. It's just kind of like passing the torch down to the next protein, but you're just passing the electrons through. And then of course at the end is where you're going to have the largest payoff of ATP that we'll talk about in a little bit. Okay, so our oxygen pulls electrons down the chain in an energy yielding tumble. So like I just said, it's kind of like if you imagine a bouncy ball going down the stairs. It's going to hit each of the stairs, and as it's going, it's going to be lower and lower energy. The energy yielded is used to help us to generate our ATP molecules. That is the fuel of our bodies. We need it to do almost everything. So the stages of cellular respiration, an overview here. So we have glycolysis is the very first, right? There's 3 overall steps that we talked about. So harvesting the energy from glucose has 3 major steps. Okay, so the first one is glycolysis. Glyco refers to sugar. Lysis means to break. The word itself literally means break sugar. Okay, well that makes sense. It's a catabolic reaction. We're going to be breaking it apart. And what are we breaking apart? Glucose, because that's the thing that we're talking about, the organic molecule that we're using. Okay, so it breaks down glucose into 2 molecules of something called pyruvate. Glucose is a 6-carbon molecule, and each pyruvate is a 3-carbon molecule. It's just like you took it and you snapped it in half. That's going to be very important later. Okay, pyruvate, oxidation, and the citric acid cycle are the next part. So you have to turn pyruvate into like another substance before it can actually go into the citric acid cycle. But that's going to be the complete breakdown of glucose. Okay, and then lastly, we have oxidative phosphorylation, which accounts for most of the ATP. Okay, that's where we're going to be generating most of it. So that's going to be through the electron transport chain. So really, I like to call the 3 steps glycolysis, citric acid cycle, ETC for electron transport chain. Those are the 3 major steps of the overall process of cellular respiration. And like I said, there are both anaerobic and aerobic cellular respirations. If we just say cellular respiration and we don't specify, it's safe to assume we're talking about aerobic cellular respiration. If I indicated it's anaerobic, then that would be a different process, which we'll talk about later. Okay, but if it doesn't specify, it's safe to say that it is aerobic cellular respiration, which does go through all 3 of these stages in order to produce ATP. So the formatting here is a little bit off, thanks textbook company. But anyway, this is showing you the color coding for the rest of the presentation. So glycolysis is going to be teal. You have our citric acid cycle that's going to be more of this like orangey salmon color. And then you have oxidative phosphorylation, which is the electron transport chain, and chemiosmosis, which is going to be more of this like light lavendery purpley color. So that's going to be true for the rest of the presentation and also the textbook. If you happen to be using the same textbook as my kids, they're using the Campbell Biology and Focus textbook. I believe that this one's like the 10th edition or something. But it's true for any of Campbell's biology textbooks. The color coding's the same. So here's an overall picture of what's happening. And like I said, the color coding here, it's nice because glycolysis is in that teal. You have the citric acid cycle that's going to be the salmon color. And then we have the electron transport chain that's going to be purple. Okay, so this is showing you the overall process. And I think that this is another really great image that you should be coming back to kind of see where everything is happening. This is showing you the overall reaction of cellular respiration, again, assuming it's aerobic, where everything is going in, coming out, being made. Okay, so the process that generates most of the ATP is called oxidative phosphorylation because it's powered by redox reactions. Oxidative phosphorylation accounts for almost 90% of the ATP generated in cellular respiration, the process of cellular respiration. So we have the 3 steps. We have glycolysis, we have the citric acid cycle, and then we have the ETC. So oxidative phosphorylation occurs with the ETC. Okay, so 90% of it comes last. So just remember that. That's at the very end. Okay, a smaller amount of ATP is formed in glycolysis and the citric acid cycle by substrate level phosphorylation. So substrate level phosphorylation does not generate as much energy as oxidative phosphorylation. And if you look back at this picture, it kind of shows you that here in glycolysis, it's substrate level, and citric acid, it's substrate level. And then in our oxidative phosphorylation step, that includes the electron transport chain and chemiosmosis, it's oxidative phosphorylation that generates a lot of ATP. That's why these little ATP clouds are small and this one's larger. Okay, so that will be something that you're probably going to be asked on your exam to know the differences between oxidative phosphorylation and substrate level. That oxidative makes more ATP. For each molecule of glucose that's degraded into our carbon dioxide that you're going to exhale and water that you're going to exhale, the cell makes up to 32 molecules of ATP. This assumes that like nothing messes up. This assumes that it's like in a perfect world, it can create up to 32 molecules of ATP. So we have enzymes that are going to help us do this, right? We've talked about this before. So this is when we have an enzyme and a substrate. You have a phosphate that's attached to the substrate. It's going to be cleaved and added to ADP in order to create ATP and a product. So that's what this enzyme is showing you. This is the reaction that the enzyme is catalyzing here. It's just speeding up this reaction, having a substrate that has a phosphate group on it, substrate level phosphorylation. Okay, it's going to donate that phosphate group to ADP, which is adenosine diphosphate, to make sure we have adenosine triphosphate, which is actual cellular energy. And then you will have a product coming out as well. Okay, so glycolysis is our first step. So glycolysis harvests chemical energy by oxidizing glucose into 2-pyruvate. Okay, so glycolysis, the word itself means sugar breaking or sugar splitting. So it breaks down glucose into 2 molecules of pyruvate. Like I said, glucose is 6 carbons, pyruvate, each one is 3 carbons. 3 plus 3 is 6. That checks out. So glycolysis occurs in the cytoplasm. I'm going to say that again. Glycolysis occurs in the cytoplasm. Didn't I say that cellular respiration happens inside the mitochondria, but then this is happening inside the cytoplasm? So like, why is that a thing? Glycolysis is special, happens in the cytoplasm. We'll talk about why it's special later when we talk about evolution towards the end. Okay, glycolysis happens in the cytoplasm, not the mitochondria, in the cytoplasm. And who has cytoplasm? Every single cell has cytoplasm. These 2 things are going to go together later. It's going to be important. Okay. So you have 2 major phases. You have the energy investment phase and the energy payoff phase. Okay. So glycolysis occurs whether or not oxygen is present. So this means that glycolysis is occurring in both aerobic and anaerobic cellular respiration, which is also called fermentation. Okay, glycolysis occurs no matter what. So this means that every organism is doing glycolysis. And like I said, every organism also has cytoplasm. So that's convenient. Does every organism have a mitochondria? No, because prokaryotic cells, which are bacteria, no, right? Prokaryotic bacteria, no nucleus, no membrane-bound organelles. So a bacterial cell does not have a mitochondria, which means that it can at least do glycolysis here, because glycolysis occurs in the cytoplasm, and it absolutely has cytoplasm. So that's very important. Okay, so we're going to be talking about the process of glycolysis. So you have the investment stage and the payoff stage that we'll be getting into. So here's the overall reaction: that you have the energy investment. You're going to be using 2 ATP, but then later you're going to be generating 4 ATP for a net of 2 ATP. Okay. So this is the investment phase. So all these little numbers, 1, 2, 3, 4, 5 that are in this picture, all those numbers represent enzymes, and all these enzymes have to catalyze these reactions. It's important to understand that if it's a kinase, you are going to be either adding or removing a phosphate from something. Okay, so hexokinase, hexo is referring to 6, so 6 carbons. Kinase tells you it's moving a phosphate group. So glucose becomes glucose 6-phosphate. That means that on the 6th carbon, we've added a phosphate, glucose 6-phosphate. Where did that phosphate come from? Well, we used an ATP to do that. So we took ATP and we made it into ADP, that extra phosphate we slapped onto the 6th carbon of glucose. So hexo 6 kinase means it moves a phosphate. Hexokinase took the phosphate from ATP and added it to glucose to make glucose 6-phosphate. This is how you should go through each one of these steps. If I do that for all of these 10 reactions, we're going to be here for like 7.5 hours. Okay. But that's how these things work. If you see something like number 2 that says isomerase, phosphoglucoisomerase, it means it's rearranging the molecules that are present here. We have glucose 6-phosphate that becomes fructose 6-phosphate. The phosphate doesn't move, but it's the arrangement of the bonds that are going to change. Isomerase is just kind of rearranging. So if it's a kinase, it's moving a phosphate. If it's an isomerase, it's rearranging. Okay. So those are the 2 really important ones that you'll see used throughout the 10 steps. This is also outlined in your textbook. Okay. Like literally step number 1 in your textbook says that hexokinase transfers a phosphate group from ATP to glucose, making it more chemically reactive. The charge on the phosphate also traps sugar in the cell. And then for step 2, phosphoglucoisomerase, it's saying that glucose 6-phosphate is converted to fructose 6-phosphate. So you do need to kind of know what's happening in these steps and what these enzymatic names are telling you. Okay, but this is the investment phase where you're putting in 2 ATP. And this is the energy payoff where you're going to be creating 4. So if you have 4 and you take away 2, you've really netted 2. Okay. So like I said, this is, if you want to know the pictures for this and this, in our textbook that we're using, the 10th edition is on page 140. Okay, you do need to know all of those little tiny steps. Okay, so after pyruvate is oxidized, because the whole point of all of this was to make pyruvate, and you see these little 2s that are in front of everything, this is saying that glucose has already been split. So when we make our G3P here, we're going to be using 2 of these because you only see 3 carbons and there's really 6 carbons. So you're going to create 2 1,3-bisphosphoglycerate. You're going to be creating 2 3-phosphoglycerates. Okay, so that's what the little 2s mean. And that's why it looks like there's like 2 cards almost, because it's showing you that there's 2 of them. Okay, so after our pyruvate is oxidized, the citric acid cycle completes the energy yielding oxidation of organic molecules. So basically glycolysis. It took glucose, it split it in half, and we made 2 of these things called pyruvate. And there's 1, 2, 3, right? Where we have 3 carbons. We essentially took glucose, snapped it in half, boom, we have 2 pyruvates. Okay, now what this is saying is that we're going to hand off that pyruvate to the citric acid cycle, but there's one step that has to occur in between in order to get pyruvate into a usable source here for this product. Okay, so in the presence of oxygen, that's important. In the presence of oxygen means it's in an aerobic condition. Okay, pyruvate enters the mitochondrion, so it goes inside the mitochondria. Okay, and this is going to be in our eukaryotic cells, which is animals and plant cells. Notice it didn't say bacteria because bacteria don't have a mitochondria, because we just talked about that, and that's why they only do glycolysis, and we'll get back to them in a little bit. Okay, so we have our pyruvate enters the mitochondria if there's oxygen. Okay, where oxidation of glucose is completed. So we're breaking down glucose in order to make ATP. Well, we broke glucose in half. Well, that's not ATP yet. We got to keep going. Okay, that's what we're going to do inside of the mitochondria when we have oxygen. So before the citric acid cycle can begin, we have to change pyruvate. Pyruvate must be converted to acetyl coenzyme A, which is abbreviated acetyl-CoA, which links glycolysis to the citric acid cycle. It's just kind of a rearrangement that has to happen in order for acetyl-CoA to actually be fed into the citric acid cycle correctly. So right now we're on the salmon section, which is pyruvate oxidation, and then you have citric acid cycle. So like I said, we're going to do one little tiny step before we go into the citric acid cycle. So you have pyruvate from glycolysis that has there's 2 molecules per glucose, because glucose has 6 carbons and pyruvate has 3 carbons. So you will create 2 pyruvate from 1 glucose. Okay. And then you're going to go through a series of reactions here in order to form acetyl-CoA. So this is where your carbon dioxide is actually created. Okay. And then we have our CoA, acetyl-CoA, that's going to be here, that's going to help us feed into the citric acid cycle. So notice that you have 6 carbons in glucose. When you split them in half, you have 3 and 3. That's pyruvate. 3 carbons here. You have 3 carbons. When you release the CO2, you now have 2 carbons left over. So we have a 2 carbon containing molecule that's being fed into the citric acid cycle. So the citric acid cycle is also called the Krebs cycle. They're the same thing, just like Calvin cycle and dark reactions mean the same thing. Citric acid cycle and the Krebs cycle mean the same thing. Okay, it completes the breakdown of pyruvate to carbon dioxide. So the cycle oxidizes organic fuel, like glucose, derived from pyruvate. Okay, we're going to be generating 1 ATP molecule, 3 NADH, which can later be turned into ATP, and 1 FADH2, which can also be turned into ATP per turn. So we're going to be creating ATP, NADH, and FADH2 for each one of these pyruvate that we're going to be oxidizing and breaking down. So now we are officially in the citric acid cycle. Why? Because we've taken that pyruvate, we've changed the form a little bit, we've created acetyl-CoA, which is a 2 carbon containing molecule that we're now going to feed into the citric acid cycle. So this is the overall process. Does it look like a lot? Yes. But again, all these little number 1, 2, 3, 4, all the way through 8. Okay, those are all representing different enzymes that are going to be helping to create each of these little intermediates before we get to our products. And the point of all this is to be creating these NADHs, ATP, FADH2, because what are we doing this whole process for? Cellular respiration. The point is to create ATP. So if we can use NADH and FADH2 to create ATP, this is a step in the right direction. And of course, creating ATP, that's also another step in the right direction. So we are breaking down glucose all the way in order to use the products that we're making here, NADH, FADH2, ATP, to generate more ATP. Okay, so acetyl-CoA comes in, you have a series of reactions to create citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, all the way. Okay, I don't want you to get too bogged down by all of these intermediates. Okay, but what I do want you to know is what's happening here, all of these different enzymatic reactions that we have, the purpose of all of these, like you're like, well, what matters if we have, citrate versus oxaloacetate? Who cares? Well, I mean, look at the differences in the carbons. Okay, we have a difference in the carbons. We have our products coming out that we're looking for because they're going to go on to our 3rd step called the electron transport chain, which is our oxidative phosphorylation. Okay, so the point of the citric acid cycle is to further break down our glucose molecule all the way. And then we're releasing these little energy harnessing, products along the way that are then going to go into the next stage. So again, in your textbook, it spells out what each of these little steps actually does. What I want you to know is that each one of these little steps is important in order to create our FADH2, NADH, and ATP. So the citric acid cycle has 8 steps total. How do you know? There's 8 of them. Okay, they're all numbered. Those all represent the different enzymes that are going to be functioning here. Okay, each catalyzed by a specific enzyme. So the acetyl group of acetyl-CoA joins the cycle by combining with oxaloacetate to form citrate. Do you see what's happening here? Oxaloacetate right here is going to meet up with our acetyl-CoA to create citrate. This looks very similar to photosynthesis, if you ask me. Okay, the processes are kind of similar. Okay, sorry, I went the wrong way. Okay, so acetyl group of acetyl-CoA joins the cycle by combining with oxaloacetate to form citrate. Boom, there's our first product. Okay, the next 7 steps, decompose the citrate back to oxaloacetate. Again, we're having to regenerate something here, which sounds, again, very similar to photosynthesis, making the process a cycle. So as you go through, you break down citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, back to oxaloacetate. Why? Because you're going to have another acetyl-CoA that comes in, and it needs to combine with another oxaloacetate. So this makes it a cycle. It is a cyclic function here because you have to regenerate that oxaloacetate in order to accept the next acetyl-CoA that's coming in order for us to generate all of these extra NADHs, ATP, and FADH2. Okay, so the NADH and FADH2 produced by the cycle relay the electrons extracted from food to the electron transport chain. So essentially, these were electron acceptors that have accepted the electrons, and now they're going to carry them over to the electron transport chain to drop them off. That way we can make our ATP. So during oxidative phosphorylation, chemiosmosis couples electron transport to ATP synthesis. Okay, first of all, before I go on, let's talk about chemiosmosis. Okay, so you know what osmosis is. Osmosis is the movement of water from high to low concentration through a selectively permeable membrane. We're not really talking about water here. Okay, we're going to be talking about chemicals. Okay, so specifically, we're actually going to be talking about our hydrogen that's going to be diffusing through the membrane. So a process in which energy stored in the form of a hydrogen ion concentration gradient, right? You have a gradient of hydrogen ions as they cross the membrane, and they're used to drive cellular work, like the synthesis of ATP. It's called chemiosmosis. So it's actually just referring to our chemicals, or in this case, our hydrogen ions that have created a gradient. You're pumping them on one side of the membrane, and then as they diffuse back in, it's called chemiosmosis, and that gradient is going to help us drive the production of ATP. Okay, so following glycolysis in the citric acid cycle, NADH and FADH2 account for most of the energy extracted from our food. These 2 electron carriers donate electrons to the electron transport chain, which powers ATP synthesis via oxidative phosphorylation. So the pathway of the electron transport chain, okay? The electron transport chain is in the inner membrane, the cristae of the mitochondria. Remember that it kind of looks like a bean with like a little wavy line in the middle of it? It's a double membrane organelle. So we're talking about the inner membrane of the cristae, right, inside of our mitochondrion. Most of the chain's components are proteins. So a lot of them are transmembrane proteins. Some of them are integral proteins, which exist in multi-protein complexes. So we'll look at a picture of that in a minute. It literally is just a whole bunch of different proteins that are close together in this membrane. The carriers alternate reduced and oxidized states. So these are all redox reactions that are happening as we're passing the electrons through, because they're accepting and then donating electrons, right? So they're getting them from the protein that came before and then donating them to the next protein. So the electrons drop in free energy as they go down the chain and are finally passed to oxygen. So oxygen is our last electron acceptor, forming H2O, which is called water. And in this case, we're forming water vapor that comes out of your body in your breath. So we've made it to part 3, the oxidative phosphorylation, electron transport chain, and chemiosmosis. And remember, this is where we're going to get the biggest bang for our buck. We're going to make a lot of ATP here because it's oxidative phosphorylation instead of substrate level phosphorylation, which was happening in the first 2 steps. So we have a whole bunch of these little protein complexes here. And you can see that we have kind of like it's going down in our free energy. So like I said, it's like bouncing a bouncy ball down the stairs. It starts high, ends low. So we will end in a lower energy than where we started. So this is the overall process of the electron transport chain. You just have a bunch of handing off that's occurring of all of these different electrons, right? So you know that you have NADH coming in and FADH2 coming in. They come in at different steps. That's fine. Okay, they're going to be donating their electrons to then become NAD plus and FAD, which can then go back and pick up more electrons to keep bringing them. It's kind of like a go-between, okay? So the electrons are transferred from NADH or FADH2 to the electron transport chain. They're donating them. That's what's happening here. Okay, and if they're losing, oxidation is loss, which means that these guys, all these proteins are getting reduced. Okay, so the electrons are passed through a number of proteins, including cytochromes, each with an iron atom. That's just what a cytochrome is, to oxygen. Oxygen is the final electron acceptor, and it becomes water, okay? The electron transport chain generates no ATP directly. But what does happen here? It breaks large free energy drop from food to oxygen into smaller steps that release energy in a manageable amount. We have the energy coupling mechanism here that we're going to talk about, chemiosmosis, which I just explained earlier, which is how we've pumped all of our hydrogen on one side of the membrane. And then as it diffuses back down, we're going to use that to drive the synthesis of ATP through ATP synthase. So the electron transfer in the electron transport chain causes proteins to pump our hydrogen ions from the mitochondrial matrix into the intermembrane space between the two membranes. Okay, so then our hydrogen then moves back across the membrane because we've generated a high concentration. Now it wants to go to the low concentration, passive transport. It's going to move back across that membrane, even though we just pumped it out. We pumped out a whole lot, and now we want to bring it back in through diffusion. It's going to be passing through a protein complex called ATP synthase. It's a protein complex that generates ATP. Like ATP synthesis, ATP synthase. That's why it got its name. Remember, ASE tells you it's an enzyme or a protein. Okay, so ATP synthase uses the exergonic flow of our hydrogen ions to drive phosphorylation of ATP. So this is an example of chemiosmosis. It's using the work, it's using the energy of our hydrogen ion gradient to drive cellular work. Okay, so hopefully the third time through that made a little bit more sense to you. That we're using this gradient to help us. It's literally like a protein that spins. We're literally using those hydrogens to help us spin our protein to create ATP. So here's what the overall picture looks like. Okay, you have our hydrogens that are on the outside that are coming back in. They're in the intermembrane space, that orange part, and they're coming back into the mitochondrial matrix. So you have our hydrogen ions that are flowing down their gradient. They're going to enter a half channel in the stator up at the top, which is anchored into the membrane. Then we have our hydrogen ions that are going to enter binding sites within the rotor, which is going to be changing the shape of each of the subunits so that the rotor will actually spin, like I said, within the membrane. You can see that there, the arrow is trying to show you it's going to spin. Each of our hydrogen ions makes one complete turn before leaving the rotor and then passing through a second half channel in the stator into the mitochondrial matrix. So it's actually crossed that membrane. And the spinning of the rotor as each one goes through causes an internal rod to spin as well. And the rod actually extends like a stalk, like into like the knob that's below it, which you can see there. Which is held stationary by part of the stator. And then turning the rod activates a catalytic site in the knob that produces ATP from ADP and inorganic phosphate. So essentially, as all of our little hydrogen ions are coming through, they're causing the rotor to spin, and it's going to help to activate the catalytic site in the knob, the lower region, that's going to take ADP, combine it with a phosphate, and create ATP. So this is ATP synthase is the name of this little engine here. And it's like a molecular mill. It's constantly producing ATP as long as we have generated that hydrogen ion gradient. Okay, so the energy stored in our hydrogen ion gradient across the membrane couples with redox reactions of the electron transport chain to ATP synthesis. So the hydrogen ion gradient is referred to as a proton motive force, which emphasizes its capacity to do work because H plus is a proton motive force. It's helping because it's mobilizing and it's creating energy. There we go. Okay, so that whole process is chemiosmosis, where we have the chemicals called hydrogen ions coming down, kind of like osmosis, moving from high to low concentration. It's literally pushing back through the membrane to drive that rotor in order to produce our ATP. So that's our last step there. That's where we get most of our ATP from. So here's another picture of what's happening. You have the electron transport chain and then chemiosmosis happening last, okay? But the whole thing is called oxidative phosphorylation. So the electron transport chain is when we had all those little purple multi-protein complexes where you had the NADH and the FADH2 that come in and they're donating the electrons. You can see that here, carrying electrons from food. So we made these things in the citric acid cycle by breaking down pyruvate into acetyl-CoA and then going all the way through and regenerating our oxaloacetate, right? So then we can do the next pyruvate that gets turned into acetyl-CoA. We're making these NADH and FADH2s during that process, which bring and drop off electrons here. And then our electrons kind of flow like through all of these different protein complexes, reducing in energy through each complex that they go through. But all the while, they're actually causing these hydrogen ions to move to the other side of the membrane. And then as we have a large portion of hydrogen ions built up on the matrix side, they're going to become, or the intermembrane space, they're actually going to be coming back down, driving this rotation of the rotor in ATP synthase through chemiosmosis to create where most of our ATP comes from. So this whole process is oxidative phosphorylation, electron transport chains where we're transporting electrons, and then chemiosmosis is when we're relying on that proton motive force of our hydrogen ions to drive the production of ATP through ATP synthase. So an accounting of ATP production of cellular respiration, we're going to talk about where everything is made. So during cellular respiration, most energy flows in the following sequence: glucose to NADH, to the electron transport chain, to the proton motive force, to ATP. So you know that you use glucose, you're going to break it down, and this is what you're going to be creating here. It's going to carry electrons to the electron transport chain, where we're going to be transporting just electrons through all those multi-protein complexes. And then that's going to help us build up a proton motive force on one side of the membrane. As it diffuses back down, we're going to be generating a lot of ATP. Okay, so about 34% of the energy in a glucose molecule is transferred to ATP during cellular respiration, making roughly 32 ATP. That's like our maximum, okay? There are several reasons why the number of ATP molecules are not known exactly. First of all, it can kind of vary from what you're eating to the type of organism. Also, if any of those like little reactions along the way, if something like happened to mess up, there's a whole lot of reasons why there could be a fluctuation in ATP, but like ideally, we're just going to say it makes 32 ATP. Okay, so here's our overall reaction again, and this is going to be our maximum glucose number. We're doing the math along the whole way. So we have our 3 steps of cellular respiration. We have glycolysis, we have the citric acid cycle, and we have the ETC, which we're going to call that whole thing oxidative phosphorylation. So remember that these 2 steps are substrate level phosphorylation because we're just dealing with phosphates on substrates, and over here it's actually oxidative phosphorylation. Okay, so glycolysis, we put 2 in, we make 4, that's a net of 2 ATP. In the citric acid cycle, remember that you're going to make NADH, FADH2, but then also 2 ATP because you're going to make it turn twice for each one glucose molecule, because you're going to put 2 pyruvate and then they become 2 acetyl-CoA, and then each one of those acetyl-CoA needs to go through that cycle. Okay, and then we're going to create our NADH and FADH2 that are going to go into oxidative phosphorylation, the electron transport chain, and chemiosmosis to generate about 26 or 28 ATP with the functionality of that ATP synthase that's spinning to create ATP. So 2 plus 2 plus 26 or 28 is roughly 30 to 32 ATP per 1 molecule of glucose. It's a pretty efficient process. It makes a lot of ATP for 1 glucose molecule, and you consume a lot more than 1 glucose molecule at a time. Remember that the energy can come from stuff that's not glucose. Of course, if you eat fat, if you eat protein, you're just not going to be entering at, the first step of hexokinase in glycolysis. You might be entering somewhere else into the overall cycle, but having glucose actually goes through every single one of the steps, the 10 steps of glycolysis. And then you have the 8 steps in the citric acid cycle, and then the electron transport chain and chemiosmosis, right? It's the whole thing. Okay, so one molecule is going to generate 30 to 32 ATP for us. The alternative here is fermentation and anaerobic respiration. So remember that this reaction starts with glycolysis, but then it moves into, in the presence of oxygen, it's going to move into the mitochondria. Okay, this is aerobic cellular respiration. In fermentation or anaerobic cellular respiration does not have oxygen. So most cellular respiration actually requires oxygen to make ATP. Okay, without any of this oxygen, the electron transport chain will cease to operate because that's the final electron acceptor, and there's nothing there to accept electrons. It's not going to work. Okay, in that case, glycolysis couples with fermentation or anaerobic respiration to produce ATP. So we're going to take a look at that process and how it's different from our ideal situation of aerobic cellular respiration with oxygen. So anaerobic cellular respiration uses an electron transport chain with the final electron acceptor that's not going to be oxygen. For example, we could use sulfate. There's no oxygen available. Well, then you can't use any oxygen. That makes sense. So fermentation uses substrate level phosphorylation instead of an electron transport chain to generate ATP. So you know that substrate level phosphorylation is really inefficient compared to our oxidative phosphorylation. But if that's not an option for you, then you have to just do substrate level phosphorylation. So the different types of fermentation. Fermentation consists of glycolysis plus reactions that generate NAD+, which can then be reused by glycolysis. So you have 2 common types are alcohol fermentation and lactic acid fermentation. So like when you're running and you might feel burning in your legs, or you're lifting weights and you feel burning in your arms, that's actually lactic acid. So does that mean that there's no oxygen in your body? No, because then you'd be dead. But it means that maybe your muscles aren't getting quite enough oxygen. So some of your cells are starting to do lactic acid fermentation. So a byproduct here is lactic acid, and that's what causes the burn. We also use alcoholic fermentation to create alcoholic beverages through fermentation processes like beer and wine, because alcohol, ethanol, is a product of that. So in alcoholic fermentation, pyruvate is converted to ethanol in 2 steps. The first step releases carbon dioxide from pyruvate, and the second step reduces acetaldehyde to ethanol. And then alcohol fermentation by yeast is used in our brewing, winemaking, and baking processes. And actually, like when you're making bread, you use yeast, and yeast do alcoholic fermentation. Does that mean you're going to get drunk off of eating bread? No, because you're actually cooking it in the oven. It actually burns off any of the little tiny amounts of alcohol that are being created, right? But all the holes in your bread are actually just where carbon dioxide had accumulated from that little tiny organism that was doing alcoholic fermentation. So that's pretty cool. Here's the overall process for alcoholic fermentation and lactic acid fermentation. You see that they both utilize glycolysis, because glycolysis happens in the cytoplasm, and all cell types have cytoplasm. So there is no mitochondria being utilized here. Whether or not it's in the cell, cool, whatever. It's not being utilized because there's no oxygen or not enough oxygen present. So you can see that in alcoholic fermentation, you're going to be generating 2 ethanol at the end. And in lactic acid fermentation, you're going to be generating 2 lactate at the end. And that's what causes the burning sensation in your muscles, because we use lactic acid fermentation when there's not enough oxygen present for all of your cells to be doing efficient cellular respiration. But why do we do this process? It hurts us. Why do we do it? Because your cells still need energy. Like, even if it's not a lot of energy, because you're only making 2 ATP here versus the 30 to 32 that you could be making if you had enough oxygen, right? But if your cells just stop making any ATP, then they're going to stop functioning, period. right? So even though lactic acid fermentation is not ideal because it makes a very, very small amount of ATP, well, we still need ATP. So some ATP is better than zero ATP, which is why your body switches over to this process. And for things like yeast cells, they're very small. So having this 2 ATP as its sole source of energy, that's not like the worst thing in the world for it, right? For us, if all of our cells only made 2 ATP at a time from consuming glucose, we'd be in some trouble. We're very large organisms that are complex, and we've got all these different organ systems trying to keep us in homeostasis all the time. We would probably not survive on that, right? But a little yeast cell or a bacteria cell, they're going to be able to function on just 2 ATP because they don't have 12 organ systems constantly trying to function to keep them alive, right? So it makes sense that in these smaller, microscopic, less advanced, less complex organisms that they could function off of fermentation. Also, just their location might require that as well, right? But we as human beings, we would need to utilize more ATP in order to keep ourselves functioning. So this is kind of like a last resort sort of thing. Not an ideal situation, but a situation to at least be generating something. So in lactic acid fermentation, pyruvate is reduced by NADH, forming lactate as an end product with no release of carbon dioxide. Lactic acid fermentation is used by some fungi, bacteria, and also we use it to make cheese and yogurt. Lactic acid fermentation makes cheese and yogurt. It's kind of why they have that unique flavor that they have. Human muscle cells use lactic acid fermentation to generate ATP when oxygen is scarce. Like I said, it's kind of like a last resort. Like, hey, we still need to make ATP. Maybe it's not the ideal situation because we're only getting 2, but we're only doing glycolysis. It's just one step, really. Okay, comparing fermentation with anaerobic and aerobic respiration. So they all use glycolysis. Like I said, every single thing uses glycolysis. They're going to net 2 ATP from that because you have the input, the investment, and the payoff. So you put in 2, you make 4, you have net 2. Okay, to oxidize glucose and harvest chemical energy from food. In all 3 of these processes, NAD plus is the oxidizing agent that accepts electrons during glycolysis, which again is our first step that happens in the cytoplasm. The processes have different final electron acceptors and organic molecules such as pyruvate or acetaldehyde in fermentation, and oxygen in cellular respiration, regular aerobic cellular respiration, because we have oxygen, right? And then in cellular respiration, you're going to produce, in aerobic cellular respiration, you're going to be producing 32 ATP per glucose molecule, but in fermentation, you're going to be producing 2 ATP per glucose molecule. So it's a lot less efficient, but like I said, depending on the kind of organism that you are or the surroundings that you're, living in, you might not need the 32 ATP. We do, right? We just make the 2 ATP through lactic acid fermentation as kind of a last resort. We have different organisms that are called obligate anaerobes that carry out only fermentation, or anaerobic respiration cannot survive in the presence of oxygen. This is common with a lot of different types of bacteria that, like, if they actually get exposed to oxygen, they don't live, right? So we also have something called facultative anaerobes, which are like yeast and many different kinds of bacteria that are facultative anaerobes, meaning that they can survive using either fermentation or cellular respiration. So they can kind of switch back and forth between the two. And in a facultative anaerobe, pyruvate is a fork in the metabolic road that leads to 2 alternative catabolic routes. So it goes down one of 2 different pathways, as you can see here. So you'd have glucose come in, we're going to make pyruvate, and then pyruvate in the presence of oxygen can go into aerobic cellular respiration in the mitochondria. Perfect, awesome. That's going to make your 32 ATP. Or if there's no oxygen present, then we can go into the fermentation pathway and only create 2 ATP and also have that ethanol or lactic acid or other products that can be created as well. So in evolutionary significance of glycolysis, like I said, all cells have cytoplasm. All cells are capable of doing glycolysis because they all have cytoplasm. So ancient prokaryotes are thought to have used glycolysis long before there was oxygen in the atmosphere, because glycolysis doesn't use oxygen yet. That doesn't get used until we're in the mitochondria. So very little oxygen was available in the atmosphere until about 2.7 billion years ago. So early prokaryotes likely only use glycolysis to make their ATP, which they'd still get 2 ATP. So glycolysis is a very ancient process because it's thought to have derived from ancient organisms like our ancient, even like Archean prokaryotes, because there wasn't a lot of oxygen on the planet. So how could they use oxygen to generate ATP? They can't. So they would do the process of glycolysis. And that's why we think that this is like further evidence that evolution is real, because every single organism on the planet uses glycolysis, whether or not it goes on to anaerobic cellular respiration or aerobic cellular respiration. Every single thing that's alive uses the same process. And that's not just a coincidence, right? That had to be something that was conserved and passed on because it was advantageous in all species. So that's really cool that that exists. Glycolysis and the citric acid cycle connect in other metabolic pathways as well. So glycolysis and the citric acid cycle are major intersections to various catabolic and anabolic pathways. So this is talking about all the different types of fuel sources and everything that you can be breaking down in your body. So catabolic pathways funnel electrons from many kinds of organic molecules, not just glucose, into cellular respiration. So glycolysis accepts a wide range of carbohydrates. Like I said, we just use glucose because, first of all, we just talked about how it was made in photosynthesis, and then it also hits every single step. Like, for instance, if you eat fruit, you're eating fructose. Fructose doesn't enter and do all 10 steps of glycolysis. It actually only does 9 steps of glycolysis. You skip the first one, or maybe the first 2. It might only do 8 steps, right? But you get to skip some of the steps because the structure of that particular molecule is different, which is really interesting. Proteins must be digested to amino acids, and then the amino acid groups must be removed before amino acids can feed glycolysis or the citric acid cycle. So proteins take a long time to digest because you actually have to split it all into all the amino acids, and then all of those like R groups have got to be like removed before you can actually go down and break down the actual amino acid to fuel the process of glycolysis to then go into the citric acid cycle and so on. Fats are digested to glycerol that's used in glycolysis and fatty acids. The fatty acids are broken down by beta oxidation and yield acetyl-CoA that can then be fed into the citric acid cycle. An oxidized gram of fat produces more than twice as much ATP as an oxidized gram of carbohydrates, which is why the keto diet is such a thing right now, because for each gram of fat you consume, you're actually generating more ATP per gram compared to a carbohydrate. And that's it takes a little bit longer. It's supposed to last with you a little bit longer. So you're actually getting more energy from fats than you would from carbs, which makes sense why one's like long-term energy and one is short-term energy as well. So these are all the different macromolecules that you can eat, your proteins, carbs, and fats, or lipids. This is where they all enter in the whole process. So you can see that, like, if it's a carbohydrate, it doesn't have to start with glucose and then go down. It can be a different type of sugar. You can have sucrose that gets split into glucose and fructose. Glucose goes through normally. Fructose then skips a few steps and then enters where it would become phosphofructokinase by a kinase that would then add that phosphate group onto it, like, right? And then the proteins you can see over on the side, they can enter in, whether you have pyruvate right off the start, you can just enter in a pyruvate, become acetyl-CoA, go into the citric acid cycle. Sometimes your proteins can be broken down and go straight into the citric acid cycle. Your fats are going to go into both of the processes of glycolysis and be directly transformed into acetyl-CoA to then enter into the citric acid cycle. So that's why we always use glucose to explain the process, to go through the whole thing, but then understand that each of the items that you're eating contains different nutrients, and each one of those macromolecules is going to be fed into the overall process in a different place. But the result is always going to be the same, that you're going to go through the 3 steps or skip the 1 step and go through steps 2 and 3, right? And then at the end, the amount of ATP that you're going to generate is going to be different based on where you entered into the system. That's why glucose is also a good example to use, because it hits every single step. You generate the 30 to 32 molecules of ATP for that. So our biosynthesis or anabolic pathways, the body uses small molecules to build other substances. Some of these small molecules come directly from food. Others can be produced through glycolysis or the citric acid cycle. So we can also make smaller molecules that then get built together in an anabolic pathway. And here's, again, our overall inputs and outputs that we've been talking about for each of the steps, the overall process of our oxidative phosphorylation. This is our electron transport chain and chemiosmosis. And then I put this slide in at the end to kind of show you the cyclical relationship. I don't know why it's like a little bit off-centered. That's okay. Photosynthesis versus cellular respiration. Now understand the photosynthesis happens in our autotrophs. So these are things that are able to produce their own fuel sources. Every single thing goes through cellular respiration. This is probably the most missed or most, the thing that's lacking from the comprehension of this whole unit. Cellular respiration occurs in everything from bacterial cell to you and everything in between, right? So anything that is alive and requires energy is going to use cellular respiration. Now, there's different kinds of cellular respiration that we talked about. But a lot of kids, for some reason, don't understand that because something goes through photosynthesis, like a plant is going through, a sunflower does photosynthesis. True. It also does cellular respiration because once you have that glucose, that's not energy, that's sugar. You need to break that down in order to get energy called ATP, which is the whole point of cellular respiration, right? So that's the overall process. We're going to talk more about the cyclical relationship in class. I hope that this helped a little bit. I know that it was long. Thanks for sticking with me. Please go back and look at the pictures and try to explain them to yourself because that will be a really great way for you to study. Good luck with it, and I'll see you in the next video.